Good morning, everyone. My name is Gina Wen. I'm a SMID-cap biotech analyst. It's my great pleasure to introduce our first speaker, Stefan Borstel, EVP of Corporate Strategy, to give a presentation. And also with us is McDavid Stilwell, Head of IR and Communication.
Thank you, Gina. Well, good morning. My name is Stefan Borstel. I'm Head of Corporate Strategy at Sangamo. Prior to that, I was CEO of Tixel, which was sold to Sangamo in the fall. Before I go through the presentation, I encourage you to read the forward-looking statements. And assuming you've done so, let's move into the presentation. And I will start with a very simple slide. So we like to define ourselves at Sangamo as a genomic medicine company. And when we say that, we are often asked, so what does that mean? That means that we use genomic information from the patient to develop therapies. And we are quite agnostic as to the technicalities or the modalities of those therapies. We develop gene therapy, ex vivo therapies, and in vivo therapies. And if you look at this slide, and we start on the left side, with the, let's say, the less difficult part of what we do. This is gene therapy. We have today two products. One lead product, which is recruiting in clinical trial in hemophilia A. This product is partnered with Pfizer. We are conducting the phase 1/2 trial. And based on what we will observe from this trial, Pfizer is supposed to take over and go into pivotal trial. The second product that we have in this pipeline is a product that we developed in Fabry. This product has just obtained an IND, and we are expecting to initiate clinical trials later this year. If we move to the center of this slide, you have the ex vivo or gene-modified cellular therapy approach. And for that technology, we have multiple application. We started 10 years ago with HIV, as you know. This program is now, let's say, pursued or continued by academic partners, and we focus our current development in three different areas. The first one is hemoglobinopathies. With Sanofi, we are developing a product in clinical trial in beta thal, and they are conducting, or they are going to conduct, a trial in sickle cell disease. We also developed with Kite an allogeneic version of Yescarta. As you know, we have a strategic collaboration with Kite Gilead, and the first product, CD19 allogeneic, is expected to obtain an IND later this year. And last but not least, we are starting development in CAR-Treg, which again is the technology that I brought from Tixel, my former company. And this part of the ex vivo franchise is expected to file for a CTA later this year, so to enter a clinical trial very soon as well. On the right side of the panel, these are the in vivo applications, which are probably the most complex things of what we do, or the most difficult. As you know, we've released for the in vivo genome editing technology some clinical data recently, which led us to believe that we have something from a scientific viewpoint, but we have yet to reach a therapeutic range. And I will come back on that later during the course of the presentation. And on the far right, we have the gene regulation technology that we use for CNS. For that part of our portfolio, we have two partnerships, one with Shire on Huntington disease, and the other one with Pfizer on ALS, targeting C9orf. And we have a proprietary pipeline addressing tauopathies, for which we expect to release preclinical data during the course of 2019. With that, I will move to the portfolio. As you can see, we have what we believe an unprecedented portfolio in our space, with 6 programs in clinical development, actually. And by the end of the year, we'll have 3 more. So by the end of the year, we'll have 9 programs with clinical trial initiated. And this is the preclinical pipeline, with 3 of those programs moving into the clinical pipeline before the end of the year, as I was saying. I'm not going to speak about the recent achievement. Everybody knows that, but I'm going to focus on each of the product we have in the portfolio. The first one, because this is the one that we have had the most recent activity, we've released in world the preliminary interim data from the phase 1/2 in MPS patient with the in vivo genome editing technology. We have released data on the first 6 patients from the first 3 cohorts. We have then recruited or enrolled 3 more patients at the higher dose, so we will have a total of 9 patients from that trial, and we expect during the course of the year to release additional data, more liver biopsy, more activity level, IDS level, but also importantly, the physicians are eager to take all these patients off ERT, and we expect to be able to give more granularity as to what happened when those patients are taken off ERT later during the course of the year. We expect, importantly, for that trial to continue with a second generation of the ZFN technology. We have an IND open with this first generation of IND in ZFN, in MPS II, and we expect under the same IND, but with a different protocol, to start a trial with the second version of the ZFN, which will be, let's say, more efficient as proven in vitro, but also which will address the global population of MPS, as we had some restriction with the first generation. We were missing something like 20% of the patients, so we expect to be having an easier recruitment in this trial with the second generation of the technology. This technology should lead to data sometimes in 2020 on overall, together with the full or comprehensive view on what happened with the first generation and what we see with the second generation. We will make a phase 3 decision sometimes next year for MPS II. For MPS I, as you know, we've treated 3 patients. The leukocyte IDUA activity for those patients we are releasing well. They are encouraging, and we are going to wait during the course of 2019 to see what we can obtain from the liver biopsy from this patient, and also what do we observe when these patients are taken off the ERT, as this is what the physician expect to do with these patients. Same technology, but different disease. We are using in vivo genome editing in hemophilia B, small trial with enrolled so far 1 patient, and whenever we can, we will update you on the status of that trial. Moving to gene therapy, there is a lot of expectation today on this program. We've treated 8 patients from 4 cohorts. As we said publicly, there is an SMC that is going to meet during the spring, and the SMC will have 2 options: either ask us to dose escalate, which we will do if we ask to, or to expand at the last dose cohort with 3 more patients. Whatever the situation, whatever the outcome of the SMC, we will update the market. But of course, there will be granularity on data if we are asked to expand, because that will be the dose very likely with which we will continue the program if we decide to continue. On beta-thal, the trial has started to enroll. Patients have been dosed, and we expect to be having data during the course of the year. We expect in total to recruit 6 patients in 2019, but we probably won't wait for the 6 patients to be enrolled to update the market on what we see. The next gene therapy product to reach clinical stage, as I was saying, is in Fabry disease, which is another liposomal storage disorder disease, rare disease. Today, patient is more or less like for MPS II and MPS I. They are treated with regular injection of ERT, and there is room for gene therapy. Clearly, we have had some very interesting mice data that were published or communicated later last year, and we have the potential with this product to become first-in-class product in Fabry, definitely. But we'll start the trial later this year, and we'll probably have the first data sometimes in 2020. Moving rapidly, as time is flying, to the CAR-Treg platform, which is, of course, close to my heart. This is what I was doing before. This is a preclinical platform today, but we expect to bring that technology to to clinical development later this year. It's a very interesting science. Basically, we take the CAR technology, which is validated in oncology, but instead of inserting the CAR into regular T cells or T effector cells, we insert the CAR into a Treg. And the idea of the CAR Treg is that they will target antigen, which are not necessarily associated with any disease, but which are localized in tissue where you have inflammation, which are the symptom of autoimmune disease. So, for example, if you have a MS, for example, you have inflammation in the brain, and the idea is to find an antigen that is localized precisely in the myelin sheath to arm the Treg of the patient with a CAR that is going to target or to bind to that antigen, to inject IV the CAR Treg. The Treg have natural property that they are homing to the site of inflammation, and when they will encounter the antigen, they will activate, proliferate, and deliver their mechanism of action, which is basically suppressing the inflammation via the secretion of anti-inflammatory cytokine, but also via direct cell-to-cell suppressive activity. That's a very interesting science, never been tested in clinic, and we expect to file a first CTA in Europe later this year. That will be, again, a first. Even in China, this technology has not been tested in man yet. In parallel to the first development, which is in HLA-A2 mismatch transplantation, and I will say a few words on that later, we are developing a few programs in large autoimmune disease like Crohn or multiple sclerosis, but we've decided to go for the first in man into mismatch transplantation, as we believe this is a very, let's say, a good setting to test this technology for the first time in human. First, because the bar in terms of safety is probably lower than if we were to treat autoimmune, let's say, chronic patient, and also because it will be a very clean way to look at the local effect of Tregs at the site of the graft. And the idea is that we have a mismatch, HLA-A2 mismatch transplantation with a donor with HLA-A2 positive, who is going to give a kidney, by definition, HLA-A2 positive, to a recipient who is HLA-A2 negative. We take blood from the HLA-A2 negative patient, we select the Treg, we arm the Treg with an HLA-A2 CAR, then we inject the CAR Treg to the patient. The Treg will migrate to the site of the kidney, which will be the only site of inflammation in the body, but also the only site where there will be HLA-A2 positivity. The CAR Treg will bind to the HLA-A2 antigen, will be activated, will proliferate, and will deliver their local mechanism of action. And the idea of that is to possibly induce tolerance so that we can take patients off their immunosuppressive regimen. And if we achieve that goal, then that will give us a lot of lessons to apply in autoimmune disease, where the bulk of the market for that technology is. I think I'm done, Gina, with the presentation, so we can probably move to a question, unless you want me to insist on anything. Good. Okay.
Yeah. Well, thank you very much, Stephen, and also McDavid. So maybe I will start, with the clinical data. I know you reported data earlier last year, several programs, MPS II, one. So I'm just wondering, any update on MPS II in terms of, additional data from the 3 patients in the expansion cohort?
So over the course of this year, we'll be collecting longer-term data from the first 6 patients who've been enrolled, but also from the 3 extension cohort patients who are enrolled at the high dose. And throughout the course of the year, there'll be opportunities to communicate the results of liver biopsies out of the patients from those cohorts who are eligible for liver biopsies, and biochemical data, as well as, very importantly, the ERT withdrawal experience.
So any particular timeline you can share with us?
Well, first of all, you have to let the data mature, and especially with the 3 extension cohort patients, they were dosed in December and January, I believe. And so there's a period of time that has to, where the data have to mature and the effect has to stabilize. At that point, then the ERT withdrawal can begin. And that happens in a conversation between patient and physician. But we're confident that over the course of the year that we'll have sufficient information to communicate to the street.
So would that be after you wait until all the data mature and then share with us, or would be like a several, time point you would share with us?
Our approach to data disclosure now is to wait until we have sufficient data that it's interpretable, so that we can then tell a story that makes sense to the street, rather than pegging to a specific moment in time. So let's wait and let the data mature, and then we'll come to you with information that hopefully will tell a story that everybody can understand.
So like based on your experience, how long do you think that will take, for the data to mature? Like, are we talking about 6 months or?
ERT withdrawal, patients can begin to do it after about 3 months, and the liver biopsies happen after about 6 months.
Okay. So we're talking about 6 to 9 months.
So throughout the course of the year, there'll be opportunities for data disclosure out of the programs. Yeah.
Okay, great. Thank you. And then, the other one is, based on your animal data and the clinical data, is it fair to say so far, the current cohort 3 editing efficiency for MPS II is below 1%?
So first of all, we haven't disclosed the liver biopsy data out of any of the high-dose cohort patients, and so we don't know what those are at this point. But the way that Sandy talks about editing efficiency in these studies is that the most important thing is that we have sufficient IDS production that we're able to take patients off of ERT and that they can remain off of ERT. Once we've arrived at that level of IDS production, then we'll be able to backward calculate into what the optimal editing efficiency is. Without knowing the outcome, it's difficult to say what level is actually appropriate. So
I think based on the animal data, I think my understanding is like maybe a few percent editing efficiency actually lead to pretty meaningful the enzyme activity. So just wondering, because based on the clinical data, what we've seen so far, we were not able to detect a very, like above threshold the enzyme activity. So just wondering, just based on the animal data.
So we think we're close. We had patient 6 who had a substantial increase above baseline. That effect was lost due to a transaminitis, but we believe that we're close to a point on the curve where we're going to get an effect in IDS production.
Okay, and then which means, so were you planning to do the future dose escalation for this, or we will completely move to the generation 2.0?
No, we are going to move to 2.0.
Okay.
We are first going to observe in the 3 patients of the expansion cohort at the same dose as patient 6, what we see, and we are going then to move to 2.0, but we are not going to further dose escalate with 1.0.
Okay. So regarding 2.0, when we look at, I think you shared with us the exactly, improvement you made, right? So to me, it looks like mainly is try to improve the efficiency of the transcription or translation of the transgene, but not too much done in terms of improve the transduction efficiency into the liver cells. And we understand that was, to me, I think that's a key question, because you do have 3 vectors wanted to get into the same cells, right? So anything, you've done or you are thinking that you could do to improve that step?
Yeah, well, we are working permanently on improving vectors as well. We are also working on LNPs, and so far we need to wait and see what we have with the 1.0 and the 2.0 version before we do any further improvement to the product. But it's not impossible that in the future we may bring, improved vector, for example. We've published, by the way, recently in Cell, like 2 or 3 weeks ago, some preliminary works on LNP, and they seem to be very effective with ZFM.
Mm-hmm. So regarding LNP approach, can you share with us a little bit more on, the potential, like the efficiency or whatever you've seen so far in the animal, and then when will you be able to share with us the data?
Well, we've made a publication recently which is showing like 90% close or ish efficiency, but you will need to wait to see more, granularity in terms of animal data.
Okay. And then, for ZFN, 201, 2.0, like second generation, do you need to file a new IND in order to start the MPS II and I clinical trials?
So the agency is allowing us to file a new protocol underneath the existing IND.
Okay.
So manufacturing has begun, and we expect that we'll have that study, that next generation study, initiated this year in time to give us the data that we need for a phase 3 decision in 2020.
That's great.
Yeah.
So from now until the time you can do the clinical trials, can you walk us through the steps you need in order to reach that step?
Sure, to complete manufacturing, to finalize the protocol, to get the IRB sites and the sites up and running, and then to enroll the patients. And we're confident that that is all going to happen this year in a time frame that allows us to make that phase 3 decision next year.
Yeah, that's really the most important catalyst or milestone is for that program is when can we make that phase 3 decision. And that will be, as I was saying earlier, next year based on 1.0 plus 2.0 data. So we are going to initiate the trial this year, and you will have to wait a little for us to be in a position to make that call, which, again, for that program is the next most important catalyst. Can we go into phase 3 or not?
I see. So, what would be the gating factor for you to decide, phase 3 decision? And also, does that incorporate any feedback from FDA, what they are looking for as well?
Of course, there will be end of phase 2 meeting with FDA, but at the end of the day, what you want to see with these patients is that if you can take them off ERT, that's the ultimate endpoint. But we are not going to be more precise than that at this stage.
Okay. So, when we're talking about off ERT, are we talking about permanent off 100%, or is it like a percentage, certain, like a rescue, like a...
We cannot be specific, but the idea of what we are providing is to provide a cure. So when you provide a cure, you don't need treatment.
Is 100% off, right? Okay. So I think that we have a few more minutes. I wanted to ask about beta thalassemia sickle cell. I think that would be also very exciting, later this year. Maybe, any update on enrollment status? And then also, have you already dosed or infused, any patients already? And our understanding is that it should be at least 2 patients enrolled.
As I was saying earlier, dosing has started. We are not being more specific than that. We still believe we are in front, in this space, in beta thal for, gene editing technology, and we will update the market in 2019. We are going to recruit 6 patients, but probably we are not going to wait for the 6 patients to be, dosed for them to generate data for the market to be updated on that program, which is a very important program to us, but also to Sanofi.
Okay, so 6 patients. Can you walk us through, the thought process, like when you will present the data? Is it realistic for us to expect you to present the data at the ASH this year?
We are not being specific. But ASH is toward the end of the year, so yeah, you can guess that's a realistic expectation.
So that's a possibility. Okay. And then, is there anything, is it purely your decision, or you have to discuss with Sanofi to make a decision when to present the data, or how much data is ready to share?
No, we have joint steering committee with Sanofi, so everything we do, everything we see, they know it and they see it. And of course, we are not going to communicate anything without, letting them know, but we are in control until they take control of the program. So. But we are not going to do anything that they will not like. We are in relationship with our partners in general, and with Sanofi in particular, so we are not going to create any difficulties in that part.
So lastly, just quickly on hemophilia A.
Yeah.
So just you already dosed the 4 cohorts, right?
Yeah.
So what would be the gating factor to dose higher? Is there any like threshold you have that you have to reach in order to dose higher?
So the Safety Monitoring Committee will meet in the spring, and they'll review the accumulated data, and of course they'll look at safety and at efficacy, and then they'll make a recommendation to the steering committee about where we go next. But we're confident that we're going to have data in 2019.
Okay. So like say if there is no safety issue, will you continue those higher, or will you have say a threshold in your mind is like as long as we hit 30 to 50% or something, like I'm just throwing the numbers there. So like if you hit that, we will stop, or you would just, as long as data monitor committee, safety committee comment, okay, they're safe, you can continue those higher, you just continue to go up.
Sure. We and Pfizer have a very clear idea of what good looks like here, and that'll of course also be a determining factor of the next step for this program.
Okay.
Yeah.
Okay. Thank you very much.
Yeah, thank you.
Thank you.
Yeah, and then we have a breakout session, I believe is in Ponciana 2.